to meet with other people. All right, so I guess these guys are in control. Let him.
Shall I try sharing my screen? I'm trying now. Yeah, it seems to be working. Is it okay for you? Can you see my? Okay. No, that's fine. Okay. Wow. It's sent directly to Russia. Wow. Ah, okay. Cool. Yes, with yeah, yeah. Yeah, yeah, thank you. And sorry for the misunderstanding, but that's fine. Yeah, which is better. Yeah, indeed. But yeah. Mm. Ah, okay, but I don't see people. That's normal. No, no, that's fine. That's fine. Okay. No, that's fine. Okay. Oh, yeah, I can see. Yeah, I can see that there are 24 people.
All right.
Yeah. Okay.
Okay. Welcome everyone. Let me just start by a brief introduction of Claire Sergent, today's speaker of our Swartz Seminar series. Claire did her PhD first. She did her studies at École Normale Supérieure in biology. And then she spent a year at UCL in London with John Driver. And I think perhaps that's when she started to become interested in cognitive neuroscience. Claire did her PhD with Stanislas Dehaene in Paris, and then 2-year postdoc again at UCL with Jehan Rees. In 2012, she started her own lab back to Paris. Now she is professor at Université de Paris, Paris University. And Claire's work has been remarkable, especially focusing on the brain mechanism of consciousness. So she uses a combination of tools, including experimental psychology, human ECOG recording, MEG, and functional MRI, as well as computational modeling. Not only she look at healthy subjects, but also she does really very interesting work on people, say, with minimal consciousness, because of some damage or some illness. So we also actually collaborated a bit, and I had a wonderful time interacting with her when I spent some time in Paris in my sabbatical year. I think it's really a very interesting to see how care combine all those tools and collaborate with people try to understand and kind of uncover the mystery of consciousness in the brain. Thank you for joining us, Claire. Please go ahead with your presentation.
Yes. Well, thank you very much for having me, for inviting me. So as I was saying, I wish I was in New York, but I hope I'll be able to make it another time. It's really cool to be talking to this this audience from a wide spectrum, and especially people from computational neuroscience. As you were saying, I'm a biologist but indeed, as I grow up in what I'm doing, I'm increasingly interested in trying to put my interpretations into a test by modeling. So I'm coming to you as a neuroscientist, very curious of what you think of the models. So in this presentation, I'm going to concentrate on the brain dynamics associated with conscious processing, and I'm basically going to tell you a story about how I became increasingly convinced that there are special dynamics associated with conscious processing, and that this might be very useful as a signature of conscious processing, or I hope it can be useful. But first, let me present you some of my collaborators on this project and my current team. So, because obviously all this work is a collaborative work, and now let me tell you a bit more about what I mean about conscious processing, or I'll be using the term conscious access. So obviously, conscious processing might sound like a bit complex, a complex concept, very polysemous as well, but we can identify very straightforward questions within this general topic. And here is an illustration of a very straightforward question that I'm trying to resolve with my research by showing you a little demo. Maybe you know motion-induced blindness, maybe you don't. So let's make the demo. I'll ask you to fixate at a little green dot here that is going to flash, and you just fixate at this little green dot. Maybe you want to get closer to your screen so I can see your nose. And okay, so maybe at some point you will realize that one or the other yellow dot is disappearing. Can you see that? So they are not disappearing from the screen. They are always there on your retina. They are always processed by your brain to some extent, but sometimes you have conscious access to it, and sometimes you don't. It disappears. And what this suggests is that it's not because something is processed by your brain that you're conscious of this stimulus. And then the question becomes, or at least one question becomes really clear. In this type of protocols, and I'll show you different types, what's the difference? The difference in my brain when I process something unconsciously, and when I become conscious of that stimulus, I have a conscious access to that stimulus. So what I'm doing is trying to understand this dissociation, or this difference between conscious versus unconscious processing during wakefulness in healthy volunteers, and understanding that in healthy volunteers might help us also derive neural signatures of conscious processing that we can use to help diagnose people with disorders of consciousness, such as vegetative state or manic conscious state. So I'll come back to this at the end, but a lot of what I'm going to show you today relates to healthy volunteers. Right. So what are the brain dynamics of conscious access in healthy volunteers? Let's go back to a long time ago during my PhD, when I started to study this idea using a protocol called the attentional blink, which is very interesting because so you show a sequence of stimuli, and you have to detect, for example, a number within this sequence. If you only have to detect the number, it's very easy to do. You always see the number. What's intriguing is that when you also have to detect another type of stimulus before that number, for example, you have to detect this kind of sequences, OXXO or XOOX, then you realize that for the same stimulation, you have an attentional blink on the second stimulus, that, namely that you often fail to see it. And what we've shown in behavior is that in the attentional blink protocol, for exactly the same stimulations, exactly the same person will sometimes tell us that they have seen the stimulus really fine, with a high visibility rating, and sometimes they will tell us that they haven't seen the stimulus. So already at the behavioral level, we seem to have a split between two types of trials. So that's very interesting because externally nothing changes, but for the participant, subjectively, it's completely different. And we recorded EEG activity and compared EEG activity for trials where people said that they had seen the stimulus versus not seen the stimulus, and see what are the brain mechanisms that led to these very different subjective perceptions. And here is a summary of what we found. So maybe you're not all familiar with this kind of the way of showing the data. So these are EEG topographies. So it's voltage on the electrodes, flattened with the nose pointing up here. And so positive voltages are red and negative ones are blue. And what you can observe here is that when you split trials according to whether you have seen the stimulus at the end or not, in the initial sensory processing phase, you see no difference between the two. And that's up until 250 milliseconds after the presentation of the stimulus. And then something changes where, on the trials where at the end people tell us that they have seen the stimulus. You see a succession of waveforms going on up until later than half a second after the stimulation, and it's not occurring when they haven't seen the stimulus. So this already suggests that there is a null or non bifurcation around 200 milliseconds that sometimes trigger a certain type of processing, leading to the subjective feeling that you've seen the stimulus, and sometimes not, and remaining in the unconscious processing of the stimulus. And if we try to see what happens in the cortex in these two conditions, we can try to reconstruct the cortical sources of these EEG recordings. So below here, it's when people have seen the stimulus. Above is when they have not seen the stimulus. And this is a view of the cortex from below here. And so we can see the film of what's going on in our brains when we become conscious of a stimulus or not for exactly the same stimulus. And you will see that the first activations we see are 100 milliseconds after the presentation of the stimulus. Nice occipital activations, which are as strong for not seen than for seen. Then the activations are going down the ventral stream of the temporal cortex, more on the left, because these are words. Same strength on both sides. And then watch it, because that's when something strange happens. Something seems to be hooked here that doesn't happen here. And here, then you have a wider network processing the information. You have frontal activations, reactivations in the temporal cortex, while nothing much is happening now when you have not seen the stimulus. Right, so how do we interpret that? So we interpreted that in the general framework of the global workspace theory that was first put forward by Bernard Bars, and then was refined and adapted to neural data by Stan Dehaene and Jean-Pierre Changeux. And the idea for this specific context is that when you receive this sensory input, the initial sensory processing is pre-conscious. These initial phases where we see no difference between seen and not seen trials. Well, at this phase, it's not decided yet whether you're going to become conscious of the stimulus or not. At least it's our interpretation. And what happens in this critical moment of 200 milliseconds after the stimulus might be related to the attentional system getting in and either selecting the stimulus or failing to do so. And if you are reactivating, selecting the stimulus, you might trigger this global sharing of sensory information, so stabilization and globalization of this specific information to higher level areas. That might be the basis of what we call conscious processing. And that's basically the global workspace idea. And what was interesting here is that it seemed that this triggering of the global workspace is associated with a very particular dynamics, abrupt dynamics. And so at the time we made a little model for looking at whether we could reproduce these bifurcation properties with a model connecting different brain areas from sensory areas to higher level areas, with feed-forward connections being encoded as AMPA connections and feedback connections as NMDA connections. And we could indeed observe when we simulated our attentional blink, that is stimulating a first target and then a second target, we could observe on several instances of doing the same stimulation that the second target would sometime trigger sensory activations that propagate, but then nothing much happens, and it's just a sort of feed-forward sweep of activations. And on other trials, for exactly the same stimulation, we have this feed forward sweep, and then something seems to be hooked, and there are sustained activation at the highest level that seem to come down all the way to sensory areas. So that was our interpretation at the time. And then later, other collaborators found phenomena that were similar to these all or none dynamics in other protocols, such as masking protocols. So, for example, here you see that for stimuli that are of various durations, you first have initial processing that seems to gradually increase or follow the strength of the stimulus, and then later activations that seem to be more all or none, or have some all or none components in that above the threshold of conscious processing for 50 milliseconds stimuli, you see that this late activation is triggered, while when you're below threshold, this activation is not triggered. So interesting, because we see that in the attentional blink, we see that in masking. Okay, but is it really a general signature of conscious access? Because it's only been observed at that stage for visual protocols, quite complicated visual protocols, and with sharp visual transients. The stimuli are shown abruptly. So it might be a specificity of the visual system, or it might be a specificity of the way we stimulate the system. So we really want to see whether these dynamics generalize to other modalities and simpler protocols. Another way we want to generalize this observation, or fail to generalize, but at least test whether it generalizes, is can we observe these bifurcation dynamics even in the absence of decision making? And why we want to do that is because for all these protocols, we ask people to report the stimulus. Well, we need that to sort the trials according to whether people have seen the stimulus or not. But then these late activations might be related to the decision that people make on the stimulus. So do we still have bifurcation dynamics even when people spontaneously become conscious of the stimulus without having to make a decision on the stimulus? So that's really what we tested much more recently, where we built a very simple protocol, audition around threshold, with no masking, no fast transient, and with and without a task. And more specifically, there were, in the main experiment, there were 20 participants. And more specifically, people were listening to a noise like this. And within that noise, we embedded some vowels, A or E, that were played around the threshold of the auditory threshold. Okay, at 6 different levels around the auditory thresholds. And importantly, people came for 2 sessions. In one session that we call the active session, they had to make a task on the stimulus. They had to report whether they heard an A or an E, and they had to rate how well they heard the stimulus on an auditory scale. In the passive sessions, the stimulation phases were exactly the same, but the difference was that at the end of each trial, the task was completely unrelated to the auditory stimuli. And in these passive sessions, people knew that they had nothing to do on the auditory stimulus. So, for example, in the passive sessions, at the end of a trial, you would have a quiz question, or you would have a mind-wandering question, like what's on your mind just now. Okay, and another important thing we wanted to do in this study is that we wanted to specify by a model, so just a dynamical model. It's not a neural network model, just a dynamical model, specify exactly what we meant by continuous versus bifurcation dynamics. So these are simulations. So a unimodal dynamics would refer to a neural process where activations progressively increase with stimulation strength in a unimodal manner. That is, if each dot here is a neural activation on one trial, they all group around a single mean, resulting in a distribution of activations across trial that is unimodal. Okay. We want to contrast that with this idea of bifurcation dynamics. And what we really mean by bifurcation dynamics is that around an intermediate level of stimulation, we will find a split between two types of trials. So for the same stimulation, sometimes the neural process is not triggered and activations across trials remain around baseline. And on other trials, this process is activated and we see another group of trials where activations are grouped around a higher mean. And this will result in bimodal distributions across trials. So how can we assess or diagnose which dynamics are going on in our data? Well, if we apply the classical way of analyzing neural data and average activity across trials for each stimulation strength, we'll end up with very similar data. And we can't really make the difference between the two situations. So we thought, let's look at the data a bit differently and look at variability across trials and how that changes with stimulation strength. And in that case, the predictions are very different, very contrasted. For unimodal dynamics, we predict a monotonous, either a flat variability as a function of stimulation strength, or a monotonous increase of variability, or at least with stimulation strength. For bifurcation dynamics, we predict a very peculiar burst of variability around threshold because you have two types of trials. And then as you go away from threshold stimulation, you go back to unimodal distribution of responses and you decrease variability. So we thought, as a first approximation, this pattern of variability might be a good detector of whether we are in bifurcation dynamics or unimodal dynamics. So we looked at that in the actual data. So again, it's EEG data, and let's look at active listening first. So these are the activations in response to our auditory stimuli in the active condition. So you have auditory and what we call auditory N1, so auditory activations, and then followed by a P300 or a very slow positivity here. So that's a standard activations for auditory stimuli. And we wanted to look at the dynamics of these waveforms. We can first do something very simple. Take the activity of these 9 electrodes here that capture well this initial negativity and the later positivity, and look at how mean activity evolves as a function of stimulus strength, and variability in the activity evolves as a function of stimulation strength. So for the mean, we see that indeed activity is amplified. Here it's negative activity, which is amplified as a function of the strength of the stimulus here and here. But as we've seen, this cannot help us dissociate whether we're in the case of unimodal or bifurcation dynamics. So let's look at variability across trials. So first we see a monotonous increase in the variability of activity across trials as a function of stimulation strength. And then after this 300 millisecond period, we observe very clearly, very directly, these pattern that we expected was associated with bifurcation dynamics, with a burst of variability at threshold stimulation, and then going down again when we were above threshold. So that was a first indication that indeed we had the first phase of sensory processing that follows stimulation strength, and that the second phase of processing, when you're around threshold stimulation, splitting between 2 types of trials. And we observe the same profiles when we do a more refined analysis, where instead of just having a region of interest, we use all the electrodes and do a classification analysis to summarize all the activity that is related to the presence of the stimulus. Okay, so in the following analysis, when I speak about neural activity, I will mean that the summary of all the activity related to the presence of the stimulus. And to be even more precise, what we did is that we trained a classifier to classify based on the multivariate pattern, to classify the stimulus present and very strong versus stimulus absent. And then we projected the trials for all the other stimulation strength on this generic classifier. And we used the distance of the activations to the decision plane as our general measure of how much the activity on that trial reflected the presence of the stimulus. Okay, but basically it means that it's a summary measure of the activation related to our stimulus. And so now we can look at trial by trial activity and distribution of activity across trials. So again, if we look at our models, here are the predictions we make for the distribution of activation across trials for the different stimulation strengths in the unimodal model. So we predict a shift of these unimodal distribution with the strength of the stimulus. For the bifurcation model, we predict that there will be a transition phase here for intermediate stimulation, where we have two bumps, two distribution. And here are the data we obtained on an actual subject during the late processing phase, but we obtained similar results at the group level, where we do see that for intermediate stimulation level at threshold, we seem to have two distributions contributing to our total distribution of activity across trials. Right, so what we did is to fit our two models, like the unimodal and the bifurcation models, to our trial by trial data at different time points after this presentation of the stimulus. And that's a summary of how well the unimodal and bifurcation model fit our data at the group level. And 0 is the presentation of the stimulus. And we see that the initial period up to 250 milliseconds after the presentation of the stimulus seems to be governed by unimodal dynamics, and then we switched to bifurcation dynamics. And what's interesting is that our model can allow us, if we give it the activation we observe on one trial, and the strength of the stimulus, it can give us the probability that this activation is in the high versus the low state. So it gives a prediction of whether the subject should have seen the stimulus or not. And you have to notice that here the model does not have any information about the subject's response. At no point in our analysis we inject any information about the subject's response. All we do is to see whether we see a split between different types of trials in the distribution of activity. Okay, and now based on this observation of the distribution, we make a bet on whether the subject should have seen the stimulus or not, if indeed high activations correspond to seeing the stimulus. And indeed, this bifurcation model does predict, combined with activity on one trial, predicts very well the response of the subjects in the active listening session. Right, so now, so first, it seems that bifurcation dynamics occur in this with these much simpler stimuli and in the auditory domain. So now, can we generalize to passive condition? So if these late activations correspond to conscious processing, they should remain in the passive condition, where there are some stimuli that are strong enough to become conscious, even if you don't have a task to do on the stimulus. And indeed, a lot of our conscious processing is occurring in the absence of a specific task. So if this is related to becoming spontaneously conscious of the stimulus, this should remain during the passive condition. If this is related to the task, it should go away. Okay. So what is the case? Does it remain? Does it go away? Well, neither. And that's what is most interesting. On the one hand, you do have late activations. And that's very striking, I think, because people, they are just hearing these A and E, and they have nothing to do on the stimulus. They are still processing it up to more than half a second after the stimulus, okay? But what's remarkable is that it's not a P300. It's not this the same activation as we see in the active condition. So, or one possible interpretation is that this is reflecting a process that is occurring also here, except that here you also are doing something more, which is not only becoming conscious of the stimulus, but connecting it with the task. So we can test it, for example, using cross-classification. And indeed, by performing this analysis, we can conclude that these processes are actually included in the processes we observe in the active condition. And it's more intuitive when we see the source reconstructions. So during active listening, we have a network of areas, including auditory areas, parietal, frontal areas. In the passive condition, it seems to be more restrained around auditory areas, but it seems, but that's okay, that's e.g. source reconstructions, but it suggests that we also have a network that is implicated with parietal and frontal areas. But what's very interesting is that the difference between the two makes a lot of sense. It's within executive areas and motor planification areas, okay? So one possible interpretation is that if this network, late network here corresponds to a global workspace, maybe this late network for passive listening corresponds to a global playground. So a situation where the representation is maintained and circulated within a wide network of areas, but for no particular purpose. And the difference might be connecting this core global playground or not with task-related network. And the global workspace might be this global playground that is additionally rooted to task-related network. So that's my proposition. And lastly, what's interesting is that these late activations in the passive listening condition do seem to show the marks of bifurcation dynamics. And so it means that we can read out in a trial by trial activity, we can read out these two groups of trials with our model. So can we predict based on these trial by trial activations whether the subject became spontaneously aware of the stimulus or not? Well, that's a tricky question because people do not have to report the stimulus. So what we did is we used the mind wandering trials. On a proportion of trials, after the trial, we asked, what's in your mind just now? And on some trials, people said, well, I was sleeping, or I was thinking of the task, or I was lost in my mind, or I was thinking of the sound that just rang, okay? And we thought, if this is related to becoming spontaneously conscious of the sound, then in the later mind-wandering probe, people who had this late activity should say, oh, I was thinking of the sound. And so we could predict based on this activity whether people had this in their mind wandering. And indeed, there is a good prediction based on these late activations. There is a good prediction of whether people have the sound in their mind or not. So it's less good than in the active condition, but it's still remarkable, I think, because it's really, again, reading out the category of activity in that trial just based on the general distribution of activity, and not injecting anything about the response of the subject. Right, so this suggests to me that bifurcation dynamics might well be a hallmark of conscious processing in the brain. And I do realize that it's quite late, so maybe you can tell me how much time I have left, if you don't mind, because I see it's
maybe another 5 minutes.
Okay, so let's switch. So now it's really going into the project really, and I wanted to say that I just heard I had a big grant, ERC grant, to go on with that. And the idea is really to try and experimentally characterize these global workspace and global playground using fMRI and intracranial recordings. Something that's also dear to my heart and would be interesting to discuss with you is understanding how these dynamics emerge and why, using modeling, and then obviously use this knowledge to help the diagnosis of patients with disorders of consciousness. So I'm going to show just hints of like preliminary data on these. So we already have some intracranial recording data with my former PhD student, Daphne Rimsky-Robert. So that's recording in the auditory cortex. So in the same protocol, insula, inferior frontal cortex for our different stimulation strength. And what we observe is quite interesting. In the auditory cortex, we see that we don't see this mark of bifurcation dynamics. It seems that the auditory cortex is not bifurcating. The insula is nicely showing this non-monotonous pattern of variability during the late phase of activation here. And same, even stronger for frontal cortex. So that's to be continued, but it's interesting that we do see these bifurcation dynamics and we can actually try to assess where they come from by probing different brain areas. So that's so here I made a modest contribution, but I wanted to mention that we managed to see these bifurcations occur in a realistic connectome of the monkey brain with Xiaojing Wang's group, with Ulysses Klassmann and Shen Freudist-Walsh, sorry for the pronunciation. And what's nice is that they explored the parameter space of strength of AMPA and NMDA in the feedback and feed-forward connections. And they found a sort of sweet spot for these different parameters where we do find bifurcation dynamics. And this sweet spot seemed to correspond to situations where feed-forward is mainly AMPA, feedback is mainly NMDA, and feedback is particularly strong on inhibitory populations. So finally, just to mention that we started testing this protocol in non-communicating patients. So that's the very beginning, and there are some encouraging data. So in a vegetative patient, you see that we see the brain's response function to increasing stimulation. We need much stronger stimulation than in healthy volunteers. So we see this nice increase in brain response to increasing stimulation in the mean. But here in this vegetative patient, variability just follows the mean. It's monotonous. But in this exit MCS patient, so probably a patient that is conscious, we see very different dynamics where, okay, mean increases with stimulation strength, but we see a non-monotonous profile in the variability, suggesting that maybe we have bifurcation dynamics. But that's obviously to be continued. And to summarize, so I think that there is a bifurcation between two ways of processing information in the wakeful brain that is associated with subjective report. And it seems to be general across protocol types and sensory modalities, and it's still present in the absence of report. So we need to understand why these bifurcations and what does it change to cognitive processing, and also use these bifurcation as a tool to assess conscious perception in different situations. And obviously, this is only a small part of understanding consciousness, but I think it's an interesting brick for understanding all the different flourishing aspects of what we call consciousness and we cherish. So thank you very much for listening. Sorry for being a bit long, and I'm happy to take questions now.
Thank you, Claire, for this, really quite interesting talk, especially focusing on the idea of bifurcation in time. Kevin, do you want to,
Hi.
Oh, hi, Claire.
Hello.
Can you hear me?
Yes.
Okay. Well, I put a question in the chat, but it was a great talk. I was really completely fascinated by it. I'm especially interested in that mind-wandering result that you mentioned, because it suggests to me that the global playground activation might derive from cognitive processing rather than consciousness itself. Because what you detected in the mind wandering result is that some of the subjects were thinking about the sound even though there was no task. So maybe what you're getting is the dynamics associated with thinking about the sound rather than passively being aware of it.
That's possible. But okay, so there are several elements for discussing this aspect. First, we have to notice that the activations are completely different from what we observe in when people are making a task on the stimulus.
Yes, so, and I'm very interested in that. Yeah.
Yeah, so that means that they are not doing the same thing in both cases.
I didn't say they were doing the same thing.
Yeah. Okay, I get your point. Second, they cannot anticipate that they will have the question on the mind wandering. They are involved in quiz questions, and on like 10% of the trials, we stop them and we say, what was in your mind just now? So at least this cannot be an anticipation of this question. It's the question comes afterwards. So that's another way we try to make people not anticipate a question they might have. But yeah, so that's two ways we're trying to avoid what you're saying, that people, although they are not asked specifically to report the stimulus, they are still like mentalizing the stimulus.
So the problem is what some people call the bored monkey problem, which is that even in the absence of a task, and even if they're not trying to anticipate a task or anything like that, they still just might be thinking about the stimulus just because they've got nothing else to do.
Well, in this case, yeah, I know the argument, but in this case, they have something else to do. They have a visual tasks on each trial. They have a task to make. So it's either a quiz question or speeded reaction time or mind wandering, indeed. But it's not that people do have nothing to do, and then okay, what can they do other than trying to record what's going on? No, I don't think they are bored. They are busy with something
Ah,
else.
okay, good point. All right, so maybe the issue is you haven't given them enough to do. So, because your mind wandering result gives positive evidence that they were thinking about that sound.
Yes, but then so then I guess we get back to a sort of dead end. If what else? How else would you assess whether they were conscious of the sound, spontaneously conscious of the sound? Um, if they tell you that they were thinking of something else, you think that they are still conscious of it, but just not thinking of it? I'm not sure. I mean,
Okay,
So,
I don't have a particular suggestion for how to tell.
may I seek a bit more clarification related to that? So if you um, focus on data from people who were not think about the sound, you do not see this, no monotonic dependence anymore.
If we sort those trials, okay.
Yeah.
That's a good point. Does it completely disappear
Right.
on trials where people say that they were not focusing on the so that's the opposite of the analysis that I did, and I have to look at that. I don't know, but that's a good point. Yeah. Yeah. Okay, I'll do that. Okay, so let me see. Yes. No, by Shan, do you think that the difference between I only see one by Shan. Okay, so do you think that the difference between the active and passive activation specifically represents the preparation of an action, or could it be any task-related cognitive operation? Well, here, I mean, it's
So we the audience can't see the question. So
okay. So the question is basically, is the difference between is this difference really just motor preparation? That's what I understand, versus like any task-related activation. So indeed, here it's EG source reconstruction. So we must be cautious in our interpretation. It's just striking that indeed the only difference is relates to motor preparation, but indeed I assume that it's the whole task-related network that probably makes the difference between the playground and the workspace. But here it seems to be restricted to motor planification. Oh, thank you, Martin. I have a jet lag, so I forgot. Did the EEG data show bifurcation in the passive condition? Yes, And does this match the intracranial data for auditory cortex? Okay, so EEG data does show bifurcation in the passive condition. Here, for example, we see this very peculiar pattern of variability with a burst of variability at threshold. And for intracranial data, I can show the passive condition. For the moment, I've only shown the active condition. In the passive condition, I don't have these plots, but I have these plots, and I will show you straight away. So it's yeah, it's preliminary data, so I don't have everything set up yet. But basically, in the active condition, we see a nice response of the temporal cortex, auditory cortex. We see the same thing in the passive condition, and interestingly, we see also late activations. Same in the insula, same in the frontal inferior cortex, but some frontal areas show a switch from nice response in the active condition to no response in the passive condition. So that's interesting. So it seems, I mean, so far, it seems that there are some areas of the network that switch off in the passive condition. The insula seems to be responding, but doing very different things, which I think is really interesting as well. Regarding the motivation of voluntary responses, and otherwise some frontal areas do respond and some cease to respond. Right.
Any other questions? So I guess just to share, a comment and see, Claire, what do you think? As we know more about how different subnetworks of areas are engaged in different components of cognitive process. Some are really cognitive, some are preparation for action. Then we could understand better if we do have the spatial and the temporal resolution, in your measurements. Then we could have a better understanding whether, or not, there's really a core, right, system underlying conscious axis. Does that make sense? I guess right now there's a bit of a limitation of spatial resolution from EEG, right? And a temporal resolution from functional MRI to really, instead of looking at all signals from all probes, I think in principle, in the end, we should really look at, some distinct sub-networks of brain regions.
Definitely. So, well, intracranial recordings are starting to help us do that, but it's not, obviously it's not everywhere in the brain, or we would have to have like thousands of participants. But the way I'm attacking exactly what you're saying is by now switching to fMRI with the same dynamic analysis. So the idea is that we first find the network that responds to the stimulus. We have our, either in the active or passive condition, we have our regions of interest, and within that, we look at the dynamics of each of them. So, of course, we won't know, we will have a summary of the dynamics over the course of a few seconds, but then we can match that with the temporal resolution of EEG, and also try to this I'm not sure about, but I have a hope that we might have the same subjects in fMRI and MEG and try to merge the data. But obviously the grail would be exactly that, having both temporal and spatial resolution and really see the different networks and their temporal dynamics. But that will be in 5 years.
Right. You're a big European grant.
Yeah.
I cannot resist but asking you about the subcortical structures. There's some suggestion that, say, medial dorsal thalamus actually is very important for awareness. And can you share us your thought, but also maybe with your clinical patients? Are they subjects with the thalamic damage?
So obviously, I mean, there is an interaction here. We're looking at participants that are healthy and awake, but there is an interaction of this conscious access process with vigilance state. Okay, so that's just my view, but it's a bit general that there might be subcortical structures might put the general system in a situation to be able to trigger these bifurcations during normal wakefulness and shunt this down during during completely unconscious states. That's a possibility. Now, if we look at patients with disorders of consciousness, groups like Stephen Lowry's, who really started studying these patients, said they show a dissociation between vigilance. They are awake, but not conscious. So there might be a dissociation between vigilance and consciousness. I don't think it can be completely dissociated. I think there is an enabling factor of a general state of the system that allows these stabilization, global activations to occur, and that this enabling factor critically involves subcortical structures.
So I see there's another question here. Kevin, do you want to?
So can you repeat that? So okay. So maybe if I rephrase, that would mean in the passive condition, the stimulus is competing with default mode network, or I think it's okay. Yeah. Okay. Well, if that's what you meant, I think it's, yeah, that's exactly what is occurring. In passive listening, the stimulus is competing. Well, in both active and passive listening, the stimulus is competing with the default mode. We know that as soon as we don't have a task, there's this switch to the default mode. So we could say that it's the case in both conditions, but it's even stronger in the passive condition. That's probably the case. So is the switch off of certain areas due to this competition? I guess yes, but also due to the fact that you just don't have to connect the information to any task. But indeed, that's a component of what we're seeing, I think, is competing with the default mode. Also, yeah, what we see is that the threshold of the brain's response function is shifted. So it means that people hear less well in the passive condition than in the active condition. And that makes sense as well.
Yeah, okay, I think that's what I asked. Yeah.
Okay, cool.
Thank you.
You're welcome.
Great. If there's no more question, thank you again, Claire.
Thank you.
It was great to see you.
Great to see you too.
Bye-bye.
Goodbye.
Thank you, everyone. Bye-bye.
Bye.